The nucleus is one of the most vital organelles in eukaryotic cells, often referred to as the control center of the cell. It houses the cell's genetic material and coordinates critical cellular activities such as growth, metabolism, and reproduction. This comprehensive note explores the nucleus in detail, covering its structure, functions, and significance in cellular biology. 1. Introduction to the nucleus. The nucleus is a membrane-bound organelle found in eukaryotic cells. It was first described by Robert Brown in 1831 and is now recognized as the repository of the cell's genetic information. The nucleus regulates gene expression, DNA replication, and RNA synthesis, making it essential for cellular function and inheritance. 2. Structure of the nucleus. The nucleus has a complex structure, with several key components. A. Nuclear envelope. The nuclear envelope is a double-layered membrane that separates the nucleus from the cytoplasm. It consists of an outer membrane, continuous with the endoplasmic reticulum, and an inner membrane. Nuclear pores embedded in the envelope regulate the transport of molecules between the nucleus and cytoplasm. B. Nuclear lamina. The nuclear lamina is a mesh-like network of proteins, lamins, located beneath the inner nuclear membrane. It provides structural support to the nucleus and plays a role in chromatin organization. C. Nucleoplasm. The nucleoplasm is the semi-fluid substance within the nucleus, similar to the cytoplasm. It contains chromatin, the nucleolus, and various proteins involved in DNA replication and transcription. D. Chromatin. Chromatin is a complex of DNA and proteins, histones, that forms chromosomes. It exists in two forms: euchromatin, loosely packed, transcriptionally active, and heterochromatin, densely packed, transcriptionally inactive. E. Nucleolus. The nucleolus is a dense region within the nucleus where ribosomal RNA, rRNA, is synthesized and ribosomes are assembled. It has three regions: the fibrillar center, dense fibrillar component, and granular component. 3. Functions of the nucleus. The nucleus performs several critical functions. A. Storage of genetic material. The nucleus stores DNA, which contains the instructions for protein synthesis and cellular activities. B. Gene expression. The nucleus regulates gene expression by controlling the transcription of DNA into mRNA. Transcription factors and epigenetic modifications, e.g., DNA methylation, histone acetylation, play key roles in this process. Sensory DNA replication. The nucleus ensures accurate replication of DNA during the cell cycle, enabling the transmission of genetic information to daughter cells. D. Ribosome biogenesis. The nucleolus synthesizes rRNA and assembles ribosomal subunits, which are exported to the cytoplasm for protein synthesis. E. RNA processing. Pre-mRNA undergoes processing in the nucleus, including capping, splicing, and polyadenylation, to produce mature mRNA. F. Nuclear transport. The nucleus regulates the import and export of molecules, e.g., proteins, RNA, through nuclear pores. RAN GTPase and carrier proteins, importans and exportins, facilitate this transport. 4. Chromatin and chromosomes. Chromatin is the dynamic form of DNA within the nucleus. It undergoes structural changes to regulate gene expression and facilitate DNA replication. A. Chromatin structure. DNA wraps around histone proteins to form nucleosomes, which coil into higher-order structures to form chromatin. B. Euchromatin versus heterochromatin. Euchromatin is loosely packed and transcriptionally active. Heterochromatin is densely packed and transcriptionally inactive. C. Chromosome formation. During cell division, chromatin condenses into visible chromosomes, ensuring accurate segregation of genetic material. 5. Nucleolus and ribosome biogenesis. The nucleolus is the site of ribosome production. A. Nucleolus structure. The nucleolus has three regions: fibrillar center, dense fibrillar component, and granular component. B. Ribosome assembly rRNA is synthesized in the nucleus and assembled with ribosomal proteins to form ribosomes. Ribosomal subunits are exported to the cytoplasm through nuclear pores. 6. DNA replication. DNA replication occurs in the nucleus during the S phase of the cell cycle. A. Helicase action. DNA polymerase adds nucleotides to the growing DNA strand, with leading and lagging strands clearly depicted. C. Okazaki fragments. Okazaki fragments are synthesized on the lagging strand and ligated by DNA ligase. 7. Transcription and RNA processing. The nucleus is the site of transcription and RNA processing. A. Transcription initiation. RNA polymerase binds to a promoter region and unwinds the DNA. B. RNA polymerase elongates the mRNA strand as it moves along the DNA template. Century RNA processing. Pre-mRNA undergoes capping, splicing, and polyadenylation to produce mature mRNA. 8. Nuclear transport. The nucleus regulates the movement of molecules between the nucleus and cytoplasm. A. Import and export. mRNA, ribosomal subunits, and proteins are transported through nuclear pores. B. Role of RAN-GTPase. RAN-GTPase regulates the directionality of transport by hydrolyzing GTP. C. Carrier proteins. Importins and exportins facilitate the transport of molecules. 9. Gene regulation. The nucleus plays a central role in gene regulation. A. Transcription factors. Transcription factors bind to enhancer or promoter regions to initiate or repress transcription. B. Epigenetic modifications. DNA methylation and histoneacetylation regulate gene expression. C. Response to signals. External signals, e.g., hormones, trigger changes in gene expression. 10. Nucleus during cell division. The nucleus undergoes dramatic changes during cell division. A. Mitosis. The nuclear envelope breaks down, and chromosomes condense and align on the metaphase plate. B. Cytokinesis. The nuclear envelope reforms around the separated chromosomes in daughter cells. C. Meiosis. The nucleus undergoes unique changes during meiosis, including crossing over and chromosome segregation.